right, y'all. So I'd like to welcome you to the introductory comments for our study of immunology. And so today what I want to do is establish an introduction to the human immune system and kind of let you know how this course is going to move forward, okay? So first of all, as we often do when we're looking at a course, we want to know what are the outcomes? What will you eventually learn at the culmination of this course? So I want to just take a minute to go through those. They're on your syllabus, and you can certainly read them, but I want to make sure that you kind of have an idea where this course is going. So things that we're going to accomplish in the next 10 weeks include identifying the cells and tissues that are part of the typical human immune system. Okay, so we also are going to, number 2, explain the immune responses that make up the human immune system. So the first 2 bullet points are about the typical normal immune response. The second 2 involve the second part of the course, which is then turning our attention to say, okay, so if the immune response is abnormal, what does that look like and why might that occur? So we'll look at diseases and deficiency of the immune response and what the outcomes of those diseases and deficiencies are. And then finally, how might we diagnose that and how might we prevent some of the abnormal outcomes in the immune with regard to the immune system. So in addition to our course objectives for today, I want to make sure that we cover the following objectives for this time together. And so those objectives include defining the difference between self and non-self, to describe what immunological memory means, to talk to you about what is an antigen, to understand the cells that are involved in the immune response and how they communicate with one another, right? So I put the image on the right here just as sort of to kick off the concept of what are some of the threats that might in fact be problematic for the human. And those include things that you're probably pretty familiar with, right? Bacteria, viruses, non-infectious causes such as cancer. The term at the bottom is maybe one that you're not as familiar with, that term iatrogenic. It means brought forth by the healer. It's a Greek word that means essentially error, right? So healthcare providers are humans. They make mistakes, and sometimes they have outcomes that are not desirable in a patient. So an iatrogenic procedure would be something that was brought about by the healer or by the healthcare provider, right? So the immune system will respond to all of these things, and that's part of our task, is to understand how that goes forward. Let's see here. Am I not able to? There we go. Okay, so let's take a moment and think about the body, right? In addition to what I want to teach you about the immune system, there are what we will refer to as barriers to infection, barriers to trauma that exist in the human body. And they include mechanical barriers, chemical barriers, and then what we're going to call biological barriers. Okay, so on the left side of the slide, outside the body, there are hundreds of billions of bacteria, viruses, fungus, all of these things that could potentially cause disease in the human. So the first line of defense is any kind of barrier, be it skin or some chemicals that the human body produces to retard the growth of a potential pathogen, or the fact that we actually have living in and on us some bacteria that are not pathogenic. They're biological biological organisms that reside in and on us that are actually protective to us. And then once we get past those mechanical barriers, if the threat has made it past those mechanical barriers, we see 2 types of response that occur that are part of the immune response. One is called the innate response, or the innate immune system, and the other is called the adaptive response or the adaptive immune system. Now, to be clear, those two blocks comprise the entire immune response, but we can separate it into two parts. And the goal, of course, being to limit the microbes that are pathogenic that are within the body. So, as I said, there are two sort of pieces of our immune response, one that's called innate and one that's called adaptive. So let's just take a minute. We'll continue to explore these throughout the course, but just as a introductory comments, what do these mean? What do they look like? On the left side of our graph here, we have some sort of threat, and within minutes, the innate response happens. The innate immune response is immediate, and it provides protection. However, that response is, we're going to call it sort of general or generic, meaning it's going to be the same exact response if it's bacteria, if it's virus, right? It's protective, but in a really generic way. It's super important because we need that immediate response. On the other hand, if you see the second line called adaptive immunity, you note that it doesn't begin until maybe hours or days, and in some cases, with some pathogens, months later. And this response is slow because it is very tailored to or specific to a particular pathogen. So in other words, we humans can create particular responses that will only respond to Escherichia coli, E. coli bacteria, or to the coronavirus that we just dealt with over the past few years, right? And so those really specific responses have to be produced, and it takes more time to do so, right? So those are what we call antigen-specific responses. Okay, so again, we have this two-part system, which is really overall a single system, the immune system. Okay, so we're going to find out that what the immune system responds to on the surface of a pathogen is often a protein. Now, if you think about the human body, it's absolutely composed of proteins, right? So how does the immune system distinguish proteins and other aspects of the human body and human cell from proteins and other aspects of pathogens, one from the other? In other words, how does the human body know self from non-self? Well, first of all, what does it mean to be self? The immune system, quote, knows, and I'm being a little anthropomorphic here, right? Obviously, the immune system has no cognition, but we recognize our own, or our immune systems recognize our own cells because all of our cells express a receptor, which is called the MHC1 receptor, which stands for major histocompatibility receptor. And it looks like this. This is just kind of an image to give you that visual feeling, right? So on the surface of all of your cells, you have this receptor on every single one. And it is the same on all of your cells that have a nucleus. So that's just darn near all of them, with the exception of your red blood cells. They are not nucleated, okay? So by having this receptor on all of our cells, it allows the immune system to recognize that receptor and say, oh, that's a self cell, and move along and not attack that cell. Now, as we'll discuss towards the end of the course, this can break down, right? And there's many ways it can break down, but when it does, some folks will develop what's called an autoimmune response, meaning a response to self. So on that note, then, what is non-self? What is not part of the body? And how does the immune system recognize that? All of the pathogens that we might encounter, whether they're viral or bacterial or whatever else, they possess what are called pathogen associated molecular patterns. So the bacteria have patterns. The viruses have patterns. And then the human cells have receptors for those patterns, and they're called pattern recognition receptors. So a specific type of a pattern recognition receptor is called a toll-like receptor, a TLR. So for example, imagine that the picture that you're looking at here, the little rectangle, is a human cell, okay? And we have a variety of toll-like receptors on our cell and on some of our internal structures. So for example, TLR6 on that far left there, it's going to recognize parasites, okay? TLR2, kind of in the middle, it's going to recognize specific patterns on bacteria. And so this is how the, quote, immune system knows that it's a pathogen, because we possess these pattern recognition receptors that can bind to particular patterns on pathogens. So that's how the human body can determine or delineate self from non-self. So another concept that we'll continue to build on is this notion of memory. So as I shared with you in the first couple of slides, the adaptive immune response is a specific response to a pathogen, right? It's created some unique response that we're going to call antibodies to that pathogen. One of the very coolest things about the human immune system is that it can remember a pathogen for some period of time, and it's called the memory response. And that means that if you encounter the same pathogen, so the same virus or the same bacteria for a second time, your response is better because it's a memory response. So if you take a little look at the graph there, you see when you get your primary look at, or your first look at a pathogen, your initial response, even your specific adaptive response, it's good, right? It's fine. But if you then see that pathogen again, you see the same virus again, you get infected with the same virus again, the secondary response is really robust, and it's much better, it's much stronger, and that is called immunological memory. So the subsequent response is rapid and vigorous. And as it turns out, this is really the basis for the concept of vaccination, right? So as you see on the graph on the y-axis there, if we artificially expose a human to a pathogen through a vaccine, it creates an initial response. Then if that human goes out into the world and encounters that very same antigen, the response that they have is the secondary response, which is better and more robust. And so by artificially exposing a person, we are creating memory. And there's a lot of issues and things to think about around the concept of vaccination, and we'll get to that towards the end of the course, but this is the biological basis for that concept. So speaking of pathogens, we often use the term antigen in biology, and to be perfectly accurate, we should say foreign antigen, right? So what is an antigen? Well, this is a word that is a bit of it's not used in the same way every time. It is a bit of a sloppy word, I guess you could say, because sometimes we'll say a bacterial antigen, and we'll mean the whole bacteria cell. Or sometimes we'll say, oh, it's the flagella on the bacteria that is the antigen. And both of those things are true. So you can use this term to mean an entire bacteria cell, or you can use it to mean a part of that cell that is being recognized by the immune system. So at the end of the day, an antigen is something foreign that might be a bacteria, might be a virus, or it might be a part of a bacteria or a virus. It could even be a chemical, a toxin. It could be a foreign particle. Folks who suffer from seasonal allergies, they may experience an immune response to a spore, a plant spore or a mold spore, and that is an antigen as well, right? So this is kind of a generic word, but we use it a lot in immunology. So when we sort of build on that understanding, the microorganisms, so these bacteria or viruses or what, are often composed of multiple antigens. If I use that context of the word to mean a part of. So if you look at my diagram here at the top, that sort of oblong shaped thing, that is supposed to be a bacteria. And it has all these squiggly things that are supposed to represent different proteins and different structures, right? And so those are all antigens of that large bacterial antigen. So when we present material in this course, and I say, well, we create a response to this antigen, it is important, or it's interesting to imagine that the immune response is quite complex because we can create responses to all of these antigens on a single bacteria cell, right? And the part of the antigen that's actually being recognized is called the epitope. So if we enlarge this antigen and we see where the immune system interacts with the antigen, that is referred to as the epitope or the antigenic determinant. Okay, and so we'll come back to that as well as we go through the course, but I want to introduce you to some of these concepts here in our introductory meeting. These epitopes then are recognized by products that are produced by the immune system called antibodies, and they are what provide the specificity. So remember we said the adaptive immune response gives a specific immune response? Well, that's in part because of these epitopes. So if you look at our picture here, you see the epitopes are represented by different shapes. So that specificity is based on the fact that the immune system will respond by creating antibodies that are specific to these different epitopes. So you see the antibodies are represented by that Y-shaped structure. That'll be created by the adaptive immune response to specifically respond to a particular epitope, giving us that nice, really refined response that we're looking for. Oops, that's my slides there for a second. Here we go. All right. Another vocabulary word that I want you to be familiar with is this notion of a hapten, which is a small molecule that is an antigen, but it cannot be recognized by itself. It's too tiny. So in order for a small antigenic material to be recognized by the immune system, it has to be bound to a carrier, to something bigger, to make it bigger. And so the term that we use for those tiny antigenic material is a hapten. So again, this is just vocabulary that will help you as we lead into this course. All right, so I think I'll stop there, and then we'll talk about the cells of the immune system.